Hi, welcome to the lecture on EnvAdapt adaptation of animals. Now we will discuss on the neural control of the muscles, especially on the skeletal muscles, because the skeletal muscles can't move if their neurons are not telling them to do it. So we have motor neurons that are starting from the spinal cord, and the axons of these neurons are going all the way till they find the right muscle cell. And when the axons are going to and near the muscle, there will be branches. So a single neuron triggers several motor units, meaning several muscle cells. But actually, each muscle cell receives signal only from a single neuron. And in a typical skeletal muscle, there's maybe 100 or 1,000 neurons that are controlling it. And okay, if you have less frequent action potentials, there will be only twitches. But if you are increasing the frequency, then these twitches are forming, there's this summation of the force because there's all the time calcium inside the cell, and then you can get more power. And then if there is a high enough frequency, then we can get this tetanic contraction, so fully contracted cells, and this gives much higher, several-fold higher force in a cell. And depending on the cell type, the action potentials per second are different. So in the slow oxidative fibers, maybe 300 action potentials per second are needed. But then in the fast glycolytic cells, 100 action potential per second is enough. So you can control your skeletal muscle cells by having less frequent or more frequent action potentials in these 100 or 1,000 neurons. But the best way to control it, because you have so huge amount of neurons, then you can just tell that, okay, how many neurons are sending the signal. So that's why you can have this requirement of motor units. So if you are having the signal in only one neuron, then there's only few cells that are contracting. Then you are having two neurons, there's twice as much power. Then you can have 10 neurons, or 100 neurons, or 500 neurons, or 1,000 neurons that are sending the signal. And that's how you can get 1,000 times higher power in your skeletal muscles. Okay, but how these neurons can find the right muscle cell, and how the muscle cell can get the signal only from a single neuron? Well, we must a little bit figure out it on the development biology. So the motor neurons start from neural tube, and beside the neural tube, along the body axis, there are areas where there are cells gathered together, these somites, and they are very easy to find in the embryos. And these somites will form sclerotome that will, and then myotome. The myotomes will form these skeletal muscles. And when we have these motor neurons, they are splitting these sclerotomes. These sclerotomes are forming cartilage. And later on, they will form the vertebrate. So the axons are going out from the neural tube and split the cartilage and then find the muscles. And that makes sense that, okay, then between each vertebrate rudiments, we will have these motor neurons going on a typical, on certain muscles. And then this is also affecting that, okay, because these sclerotomes are split on two, and they will form these rudiments with the caudal and postal parts between these motor neurons. That makes this vertebrae a little bit more flexible. Okay, but how the neuron is finding the right muscle? Because we know that, okay, even in the leg or in the hand, we have several muscles in both sides of the bones, etc. So how the neurons can find it? Well, the idea is that on the neural tube, we have different areas. And because there are different areas, they have received different kind of paracrine factors during their development. And this causes that they have a little bit different kind of transcription factors that are affecting on what kind of proteins there exist. And because there are now differences in what kind of proteins these motor neurons have, they are then sensing different kind of attractive and forbidden chemical signals. Like, for example, they're in the medial motor column, which has 3 different limb transcription factors. It will first, the action will go directly on the limb, but then it will turn maybe 90 grade axis to go on the derma motor. And then these motor neurons are turning their axons either toward certain place or avoiding some other places. And that's why we can get axons to the core body muscles, or in the dorsal muscles, or in the ventral muscles. Okay, so therefore our central nervous system somehow knows that, okay, these cells are motor neurons to regulate this and this kind of muscle. But then how the muscle cell know that he or she will get the information only from one neuron? Well, we had to check it from the synapse, how the synapses are formed in the in these motor neural junctions. Well, the synapse forms, it's an interplay with 2 cells, and in the motor neuron, the growth cone, so the tip of the developing axon, it sends the attractive and repulsive forces, and it will be growing toward the myotome. And sooner or later, it will find it. And this growth cone will be modulated to form the presynaptic structures. But before that, in this myotome, there exist these acetylcholine receptors, but they are in random order there in the sarcolemma, all around in the cell. And when this presynaptic membrane is formed, it will release some factors called arginines and limins. And these arginines and limins are affecting on the muscle cell. And this will gather all these acetylcholine receptors in these developing synapses. But it will also affect on the gene expression in the muscle cell, in the nucleus that are near these synapses. Okay, then we can get the acetylcholine receptors near the synapse. But actually, they can be formed, these clusters, or somewhere else. But because these neurons are now starting to release the neurotransmitter, meaning acetylcholine, so this acetylcholine is keeping these receptors on the membrane. So now the muscle cells are removing those acetylcholine receptors that are useless. And after that, we will form too many synapses. So when we are having one operative synapse, it will attract other synapses there. So they can be coming from the same axon or from the different neurons. So that's why we have several synapses from different axons, different neurons formed in each muscle cell. In a typical vertebrate, there's at least from 2 different neurons to each cell during the development. But isn't that a little bit discrepancy, because there was only 1 neuron per one muscle cell? Well, the idea is now that, okay, you will have a little bit too much information. But that doesn't matter, because that's when you are still developing. Because then you are selecting the right synapses. So all but 1 axon branches will be retreated. And how they are selecting is that, okay, they are checking that, okay, are they synchronized? So if you have several synapses and they are releasing acetylcholine at the same time, then they are coming from the same cell, from the same motor neuron, and that's okay. But then if some of them are giving the signal in different phases at different time, then that's asynchronized. Then these kind of synapses will be deleted. And in some cases, there can be synapses that are completely inactive. Okay, delete them also. And the remaining axon will get the myelin sheet from these Schwann cells, and this formation will get this motor neuron a little bit faster. And that's also affecting on the retreatment of the other branches. And then we get that, okay, we are 1 neuron for each muscle fiber. But are all the muscle cells working this way? No. So in the vertebrates, we have a motor neuron that is branching and branching repeatedly to form several synapses. And that's why it regulates several muscle cells, but also the synapses can be several times in the same cell. But because they are synchronized, so that's allowed. And how it works in the synapse, it's actually not causing directly the action potential, but it's called excitatory postsynaptic potential, because it's opening these acetylcholine receptors that are allowing sodium influx, but only transiently. So it's the depolarization in the cell membrane locally. But if it's enough, then it will activate the sodium channels, and that will depolarize the cell membrane all around in the cell and cause this action potential. But in the arthropods, we had different kind of motor neurons. There can be glutamate that is causing local excitatory postsynaptic potential. But there are other neurons that are also touching the same skeletal muscle cells. And so that's why we have these multi-terminal innervation. And the interesting is that there are also inhibitory cells. So then the muscle cells are getting information that, okay, now you must contract. And then there are also getting information that, oh, no, you shouldn't contract at all. But let's go a little bit detail on that. So then it goes in the same way as in our nervous system. In the nervous system, we have excitatory and inhibitory synapses. And they can affect on the same neuron. And let's look them a little bit carefully. Okay, the excitatory presynaptic membrane is working that, okay, there will be calcium inside the cell that will release the neurotransmitter. For example, in the vertebrate neuromuscular junction, that it's releasing acetylcholine. And that will cause the activation of the receptor allowing sodium influx, but in case of acetylcholine receptor, it's also causing that, okay, the potassium can go out from the cell. But anyway, because they are going at the same speed, it causes the depolarization of the membrane, and that will cause change in the action potential tendency. So it will trigger the sodium channels, etc. And this will cause that, okay, in the presynaptic area, there will be action potential in the neuron, and then it will cause this excitatory postsynaptic potential, so the depolarization that is local, but if it's enough, then it will trigger the action potential in the cell. But in this inhibitory presynaptic synapse, there is also calcium that is causing the release of a neurotransmitter, but now beside the potassium there is chloride that is going inside the cell. And then if you have only this presynaptic, inhibitory synapse, then in the neuron there is this action potential, but now in the postsynaptic area there is a small shift in the membrane voltage, because this receptor is allowing chloride inside the cell, but it's in opposite direction than in these excitatory cells, and there's no action potential. And then this combination, is the excitatory or inhibitory larger, that regulates that, okay, will the cell contract or not. And so they are controlled usually by several neurons, and they can, in some cases, be having these two different kind of signals. And this actually causes that, okay, if there's enough excitatory signals, then there will be calcium release and the contraction. But the interesting is that in arthropods, the skeletal muscles, or these muscles that are used for locomotion, there's no action potential, because it's more like direct affecting on the calcium release, except in the flight muscles of the insects that are rapidly contracting very rapidly, and that's why there's a clear action potentials also. So even more interesting is that actually these arthropods, even the sarcomere length can be varying. So there's much more regulation. Thank you.